Yeah, welcome everybody. I'd love to introduce Eoin Brodie from Lawrence Berkeley Lab and UC Berkeley, and he looks at the intersection of soil, like plant nexus, and how the dynamic system interacts. Welcome, Eoin.
Great, yeah, thanks a lot. I appreciate the invitation, and I've really enjoyed the presentations in this series. It really is a great forum, so thank you for organizing this. I'd like to tell you a little bit about some of our work attempting to go from genomes to watersheds, and particularly trying to scale the activity of soil microorganisms from the scale at which they live, which really is the micro scale, to the scale at which we try to predict their behavior and its consequences for global biogeochemistry. So let me start with just some basics here. Like all living organisms, microbes and all life assemble across gradients of energy. The ability to assemble across those gradients is constrained, though, by resources, but also the evolutionary history. So the evolutionary history of the organisms constrains their future response to changing environmental conditions. So the availability of energy, whether that's from light or chemical energy, the availability of water, carbon, fundamentally for microbes, most of that is coming from photosynthesis through plants for soil microbes, but also from autotrophy, fixing their own carbon from the atmosphere, or in some cases from mineral weathering, and then the availability of nutrients to build cellular materials. So this is a soil microbe. When we see microbes, you're probably used to seeing microbes not in their native habitat, but in cultures or under the microscope after being grown in the lab. This is really what a microbe looks like when it's present in soil. This was probably originated as a single cell. It has replicated. It's now 2 cells. And you can see that it's encased in a matrix here, which is clay particles. So this is probably the typical mode of activity and action for a soil microbe. And we rely on soil microbes for many critical, we call them services, but this is life for a microbe. They produce soil organic matter. I'll tell you a bit more about that later. They purify our water as water moves through the soil from the surface into our aquifers, where we get our drinking water and our water for irrigation. Their role in turning over nutrients in the soil, particularly nitrogen and phosphorus, is critical for plant productivity, including our crops. They help protect against pests by producing things like antibiotics. But they play a really critical role in climate regulation by the way that they impact soil carbon cycle. And I'll talk quite a bit about that. On another aspect, though, we also rely on soil microbes for many biotechnological resources. Many of our enzymes, industrial enzymes, come from soil microbes, and probably most of our antibiotics come from soil microbes. So what do we know about microbes in general? Well, overall, across our planet, we have observed about 400,000 bacterial and archaeal species. These can be condensed into about 60,000 genera of bacteria and archaea. These are observed, and most of these are observed through using genetic tools. There's predictions of about 10 to the 12 microbial species on Earth, however. So a vast majority of microbes on Earth have yet to be discovered. However, only about 14,000 bacterial and archaeal species, that's about 3,500 genera, have been cultivated and described. So that means that the vast majority of the microbes that we have observed have never been cultivated. So that's about 3.5% if we believe the 400,000 number, or about 14 per billion of the microbes that exist on Earth have been cultivated. It gets even worse. About the vast majority of those that have been cultivated belong to just 4 phyla. These are the ones that we zoom in on because we can grow them easily. And we use these as our workhorses in biotechnology, for example. So the vast majority of microbial diversity on the planet has not been cultivated, and we've now observed them in the lab. So that means that genomes are our window. And so we know most of these organisms only by their genetic signatures. So genomes are a window into soil microbes. And this is complicated. This plot here shows that soil is the most diverse microbial ecosystem. This is a plot showing the estimated number of species relative to the number of cells in a sample. If you contrast that with the sort of the gray colors here, like the human gut, for example, this is a far less complex environment. Why? Because it's much more stable. It's a more controlled environment. Soil is a very heterogeneous environment with lots of niches at the micro scale that's capable of supporting a vast diversity of organisms. So we can't cultivate the majority of these organisms, and the vast majority of our information is in the form of genomes, and that is a complex challenge to be able to solve. It's also a complex, the complexity of this data requires approaches to simplify this and reduce the dimensionality of this data. I'll make a short statement here that how do we access this information? Well, what we do is we will sequence the DNA in soil, as much of it as possible, typically as much as we can afford to do. And then that becomes a giant puzzle. We try and take all of those short pieces of DNA and reconstruct them into genomes. So we're trying to reconstruct an entire community of genomes, many thousands of times the size of a human genome in a gram of soil. And we're trying to reconstruct that into many hundreds of thousands of microbial genomes from soil. So with that amount of information, we need approaches to try and simplify that. And we use an approach that's called functional traits. And I'll explain this. This is a really great paper published a few years back by Geneviève Lajoie and Steve Campbell. It really outlines this trait-based approach in our field of microbial ecology. We use functional traits. These are properties or attributes of microbes that can be linked to their fitness. And those could be related to the size or shape, the morphology, how they work, their physiology or behavior, and how those influence those traits, how those influence the ecological outcomes. So we use functional traits because it allows us to link genomes, the genes that contribute to the attributes, allows us to link those genes to those to traits and quantify their impact on fitness, their adaptation, and how that corresponds or influences ecosystem function. And fundamentally, though, going from genes to traits allows us to reduce the dimensions that we're dealing with. So it simplifies microbial data into more interpretable trait axes, allows us to compare across systems, and really provides a framework for us to be able to unify genomic, phenotypic, that's the expression of the traits, and ecological data. I'll say a really important part of this is using numerical models. Models allow us to synthesize the trait data and their interactions. We can use models to define functional traits, such as the sensitivity of the model to a changing value of a trait parameter allows us to put a rank order of the importance of that trait in a given context. Traits can be used as direct parameters, or we can use them to inform model structure. We can also build off the traits that we can measure to infer others using theory. I'll give examples of that. And then overall, we can use models to infer what the niches of specific organisms are from information in their genomes using simulations, and I'll give examples of that too. Okay, so microbes, really like all living organisms, are fundamentally entropy production machines. So we and microbes, we take information, use that information to process energy. That information is stored in genomes. And low entropy chemicals coming into the cell, this could be glucose that's been fixed through photosynthesis, for example, as a source of exergy, that's energy that can do work. And the path that takes is informed by evolution. So that's the metabolic pathways in the cell, the proteins that catalyze those reactions, the metabolic pathways. We store some of that exergy in cells in the form of things like ATP or maybe glucose or cellulose in plants to build structure, and they use that structure then to further catalyze the destruction of energy, producing entropy fundamentally. So these are entropy production machines. And so, as I said, the formation of these structures is guided by information, and that information are the traits that are encoded in the genome. So what sort of traits are we talking about? And I'll use an analogy here at first. So let's think about a motor vehicle. But if we want to predict certain things about the performance of a motor vehicle, we may not need to know everything about the functioning of the engine or all parts of the car. We might just need to know the engine type, the fuel tank capacity, the type of fuel it uses, what material it's made of, its size and shape, its intake systems, and maybe its exhaust systems. All of that information we could probably get from the owner's manual. And that allows us, using a simple model, we could predict the power, the weight, the top speed, the emissions, the efficiency, and maybe the maximum range of the vehicle under given conditions. So it's very similar for microbes. So microbes, we might want to know something about its maximum specific growth rate. How fast can it grow? What is the microbe made of? What size and shape does it typically form? How fast can it produce proteins? Under what temperature does it typically grow? How fast can it bring in resources? How does it acquire energy from the environment? What electron donors and acceptors might it use? Does it produce extracellular enzymes? What kinds? What are their properties? And does it produce storage compounds? That information, like the owner manual, we can get in the genome and extend that using things like allometric scaling theory and assimilation into models to predict other traits. With that, we can then predict things like biogeochemical reaction rates, the growth rate of the organism, its efficiency at using carbon, energy, and various nutrients, its other loss functions like heat dissipation or waste products, gas, for example, CO2 production and respiration. So to do this, though, we need to think of organism. We need to think at the organism scale. And there are different fields or different communities that approach modeling of microbes in ecosystems. Some folks approach this from a whole community perspective, where you consider a microbial community as a bag of genes, and then therefore a bag of chemicals as a whole community. But the trade-offs between traits occur at the organism level. An example being here a resource acquisition trade-off. So the energy per unit electron donor, that decreases with the rate of uptake of the electron donor. And you can think about that as almost like a friction term, where you have greater dissipation and greater dissipative loss and loss of heat, the greater the rate of uptake of the resources from the environment. The cells, due to evolution, have design constraints so that their size and surface area, and that provides a biophysical or a limitation to uptake of resources from the external environment. So diffusional constraints, and then the surface area relationship to the volume of the cell, that also imposes a constraint in terms of how much material a cell needs to make internally, and can it make that amount of material internally and transport the materials required effectively over time into the cell. So there's a constraint on the surface area to volume. Within the cell, then, the cell needs to make, let's call them decisions, but these are allocation optimizations. The cell has to maintain itself. So it has to allocate a certain amount of its energy and resources to just fixing and building itself over time, or replacing parts over time. Depending on the resource availability, the cell may need to make extracellular enzymes, build proteins, export them out of the cell. So that's a lost term, but with a gain function that's realized under certain situations. The cell also has to be able to build a tolerance to stressors like pH, temperature, things like osmotic stress, things like that, which soil microbes experience all the time. And the cell also has information processing trade-offs. It has to store its information. That's a function of the size of the genome. It may have to store resources. It has to ensure the accuracy of its genetic machinery, etc. as well. So all of these trade-offs occur within the cell. And so we take an approach to model these within the cell using a dynamic energy budget modeling approach. So that allows us to couple both catabolic and anabolic process, so the energy generating and the biomass synthesizing features of a cell. We couple those, as well as the carbon and energy allocation schemes. And that allows us to realize how these trade-offs occur within a cell, but importantly as well, how they're constrained by traits, and traits that we infer from genomes. And I'll give some examples of that. So that's the complexity of dealing with a cell. We have to also recognize that that cell is regulated by processes across many scales, and this is a major challenge in biology, and particularly in microbial ecology, is representing these constraints across scales. So if you start up with an H up there, the global climate, if you think about the stratification of biomes with latitude and longitude, these are basically gradients of energy and resources, energy, water, and resources coming from geology. That imposes landscape constraints. Those landscape constraints then determine the fitness, the habit of other life forms like vegetation across the landscape, depending on elevation, topography, mineralogy, etc., that determine nutrient and water availability and radiation, etc., things like that. That then determines the resources that could be made available to microbes, for example, in the soil, in the area around the root that's called the rhizosphere. This area around the root then interacts with the mineral abiotic components of soil, sand, silt, and clay, and the combination of organic matter from plants and microbes interact with sand, silt, and clay to form three-dimensional porous matrix called aggregates that create the local microenvironment that microbes find themselves in. So each of these interactions together combine to regulate the expression of microbial traits. And so you can imagine the heterogeneity that's created at planetary scale. And this explains why microbes soil microbes are really, and soil is probably the most diverse habitat for microbes and maybe life on the planet. So this is a challenge, and we're trying to build a modeling framework to try and represent not all of this at the moment, but some of this, and I'll tell you a bit about our work there. Okay, so this is we're building what's called a Genomes-to-Ecosystems framework, and I'll step through each part of this, and I'll give you examples of where we've used parts and all of this framework so far. This is a work of many individuals, and I'll credit their work as we go through each of the individual steps here. So the first step here is reading the owner's manual, reading the genome of microbes. And so what we use an approach here, this is developed by Ulas Karawas. This is a bioinformatics workflow called microtrait. And microtrait distills genomes into traits. And these are traits that we believe are important for the fitness of microbes. But of course, this is our knowledge base of what traits are important, and their genomic basis is continuously expanding. So microtrait is a series of hidden Markov models. And so those are models trained on genetic motifs from well-curated genomes with biochemical evidence of protein function. And so what we're doing is here we're identifying individual proteins within a genome that have a already diagnosed function, and then we're building an HMM approach to be able to identify and scan other genomes for those. And then we use a series of Boolean logic, a series of rules to combine multiple proteins into the presence of a pathway, for example, and then we use the presence of that pathway to infer a trait. And I'll give you examples of what we mean by that. The next slide. So there's a schematic of microtrait here. The different traits that we might represent here could be classified as life history, biophysical, thermodynamic, or metabolic. Life history traits are things that are not necessarily encoded by an individual gene or multiple genes. These are signatures across the genome that can be used as proxies for the behavior of the genome as a whole. Excuse me. An example of that might be the minimum generation time or maximum specific generation time of an organism. Basically, what the maximum growth rate an organism could achieve under evolutionary optimal conditions. And there's a signature in the genome, codon usage bias, that is very strongly correlated to the maximum specific growth rate. And that's due to codon optimization in the genome. It's a complex signature, but we can use that as a proxy for the maximum specific growth rate that a cell can achieve. We can use different amino acid frequencies to infer temperature optima. And the origin of that is differences in hydrogen bonding under different temperature regimes for protein stability. So we can read out indicators in genomes of the evolutionary optima of organisms. For biophysical traits, there's allometric scaling theory, and that relates things like the DNA volume required to build a cell, and that scales according to the minimum DNA requirement for a cell, and the maximum DNA requirement for a cell are constrained by the cell volume itself. So there's a relationship between DNA volume and cell volume that we can use we can infer cell volume. And if we assume a shape, we can then infer a surface area for that cell. The ability to use different electron donors and different electron acceptors, so what carbon source, whether a cell uses oxygen or uses nitrate or uses iron as its electron donor, we can identify that from the genome, and that allows us to understand the free energy that the microbe can harvest from its environment. And then we have strategies of microbes to use and disperse that energy throughout its cellular metabolism. We can read that from the whole metabolic map from an organism. So microtrait attempts to predict these things using genomic signatures. Then we arrive at a matrix, and this matrix would be genomes by traits. And then what we can do is we can collapse genomes with similar functional traits into what we call guilds. That approach allows us to quantify what information we are losing, how much information we retain by doing this, how much information we are losing. And then the goal is to represent guilds, not by all the representatives of those guilds, but by distributions of parameters for those guilds. And you can see that over on the far right of that heat plot, distributions. These would be organisms that generally can perform the same function, but might have different kinetic traits, like different maximum specific growth rates, might have different temperature optima, etc. So fundamentally the same chassis of an organism, but operates maybe under different conditions. So it allows us to reduce that dimensionality significantly. Okay.
Question about
yes.
kind of where that fits into a wider program. We might actually get to this, but it's what kind of activities do you have to model from biomes? Because I guess you don't care about what each species does. You care a lot about what kind of actors act in the soil. So what kind of actors should we be thinking of here?
Yeah, that's a great question. And I'll give examples of that as I go along. But briefly, so soil microbes are extremely important in transforming carbon and nutrients. And that carbon might be the polymers that are present in plant roots, for example. That can be lignin and cellulose. That carbon gets transformed by microbes using extracellular enzymes. They use some of that carbon to build their bodies. The remainder of that carbon that isn't used to build their bodies or other chemicals goes back to the atmosphere as carbon dioxide. The balance between those two, the building of the cells versus the respiration and loss of that CO2 to the atmosphere, that really determines how much carbon gets taken from the atmosphere by plants and stored in soil. So microbes are really the critical nexus, and their efficiency at which they perform that process is really essential, or it's a really important modulator on the formation of stable organic matter in soil, keeping carbon out of the atmosphere and storing it in soil. Fundamentally, that's where our fossil fuels come from as well over geological time. Microbes also produce important antibiotics in soil that can help suppress disease in soil, etc. as well. So there's a wide range of metabolisms that microbes can perform. And then I'll give examples as well as we go through. Other important greenhouse gases like methane are produced by microbes, whether that's in the rumen of a cow, or that's in a flooded field, or in a thawing permafrost. And microbes are the traits of the microbes that produce those gases determines how much of those gases are produced, when and where.
Thank you.
Okay, great. So the next step here, so we've left off here by collapsing genomes into functional guilds, genomes with similar traits, and we have parameter distributions. We use another approach called DEB microdata, and this is the dynamic energy budget model. So what we're doing now is we're feeding in this list of traits derived from microtrait into this dynamic energy budget model. and using that to predict other traits and then how those traits interact. Remember, using the whole organism perspective. So this is just a schematic, and I'll talk to this relatively quickly. We have the genome size, for example. We can predict the cell volume using that genome size, of course, with error. We use the codon usage bias to be able to predict the maximum growth rate and the translation power, so how fast an organism can create proteins. We can estimate the efficiency of that process using things like ribosomal operon copy numbers. Then to be able to infer how fast a microbe can import materials across its cell membrane. We have a cell volume, we have a cell surface area, we know how fast the cell grows, we know what it's made of, or we can infer what it's made of. So now we have a requirement of a cell to import a certain amount of material to achieve a growth rate X over a period of time Y. So that gives us the maximum flux rate through the cell, and we allocate transporter genes across the surface area of the cell. So that gives us how many transporters per substrate across that surface area. That allows us to infer what the uptake rate has to be for those transporters to be able to achieve that optimum uptake to satisfy cellular demands. So that's a new approach that allows us to infer substrate uptake kinetics. This has been a big deal because prior to this, the ability to, all of these, most of our models, as you'll see, rely on something like a Michaelis-Menten type formulation, where you have maximum specific uptake rate or enzyme velocity and half saturation constant as a constraint on enzyme activity or uptake or growth. They're very similar formulations. The parameterization of those has relied on observations from cultivated organisms. And as you remember, I started off talking about that only a fraction of a % of microbes on Earth have actually been cultivated. So we don't have observations of those. We will never have observations for most of those. So we need a theoretical approach to try and predict the substrate uptake kinetics if we're to use these approaches in our models. And that's what we've tried to build here. So again, we're using this DEB microtrait model to try and synthesize the interactions of these traits and predict things like these uptake kinetics. So I'm going to pause slightly there and see if there's any questions on this, and then I'm going to jump into some use cases. This brings us back to the fundamental role of microbial metabolism and its efficiency in soil. So let me pause there for a second, see if there's any questions, and then I'll move into some use cases.
Yeah, so should we be thinking of like whole cell models or like some approximation of that for this application, or should we be thinking more cell community kind of modeling?
Yeah, that's a good question. So I would say, so we should consider representing these interactions within a cellular basis, because there's a fundamental biophysical constraint on how these traits interact. So that's important. So when we remove that cellular context and think of a community context, we're removing that cellular biophysical constraint. I see Tom has his hand up.
Yep.
Yeah, are these dynamic models or steady state?
These are dynamic models. Yep. So let me move. I'll move on here. Maybe I should have started with this. So if you think about the carbon cycle on Earth, you probably think about all of the vegetation, and you think of the Amazon. You might think of high latitudes. You might think of permafrost as well. You'll probably think of forests and grasslands. But it turns out that there's 4 times more carbon in soil than there is in all of the vegetation on Earth. Fundamentally, most of that carbon came from vegetation at one point or another. There's 3 times more carbon in soil than there is in the atmosphere. And the soil and the atmosphere are not in equilibrium. Soil is, for the most part, a net sink. So it is absorbing more carbon than it releases, but it releases a lot of carbon. And the numbers into and out of soil on an annual basis are in the large gigaton amounts. So hundreds of gigatons into soil, about 120, I think, into soil, and about 120 out of soil, but on balance, it's a net sink, maybe 119, 118 on average. It depends on the ecosystem, it depends on the time of year, et cetera, et cetera. So soil is storing carbon, and it's storing carbon. We used to think this was the stored carbon in soil was the leftovers of decomposition of plants. So you think about all the gnarly compounds that you might have in plant roots, like lignin, etc. And there's this concept of humic acids, and these humic materials, complex materials that are the leftovers, that the things that microbes don't want to eat when plants die. It turns out that actually that's part of the story, but it really isn't the main. It's not the main feature. The main feature is that microbes can consume very simple compounds like sugars, glucose from roots. They turn the microbes are factories. They turn those into more complex biochemicals. Those more complex biochemicals, through the activity of microbes and the microbial dead bodies themselves, end up on mineral surfaces like the surface of clay. And those minerals end up encased in these aggregates that I showed at the beginning, these three-dimensional matrices that are present in soil. So the combination of microbial activity, microbial metabolism of simple compounds, interaction with minerals, and then the encasement of those minerals in a larger matrix is what makes soil a carbon sink. So this is a dynamic biological, chemical, physical set of interactions. But the modulator on this, and this is really important, is that the efficiency at which microbes take that carbon from the plant, maybe that's glucose or sugars, sucrose, or organic acids that's being released from plant roots as they grow. Microbes feed on that, build their bodies, and they build their bodies with an efficiency that's related to their traits. If they're fast growing, they're less efficient. If they're slow growing, they're more efficient. That's a simple way of putting this. The residual here is carbon dioxide. So if low efficiency organisms release more of their carbon as carbon dioxide, high efficiency retain more of that carbon in their biomass. And that builds more organic matter in soil. So microbes are a critical modulator at the center of this series of interactions. This is attractive when we're thinking about excess carbon dioxide in the atmosphere. Can we manipulate ecosystems? Can we manipulate the activity of microbes and their interactions with plants so that we cultivate or encourage the growth of microbes that are more efficient? And we've been trying to work out how this works to try and come up with strategies to improve the efficiency of microbes and the rate of organic matter production in soil. This is important also because our multiple centuries of intensive land use through agriculture and forestry, we've degraded about a third of the land globally, and we've lost about 130 or so gigatons of carbon. And about 20% of the extra CO2 in the atmosphere has originated as soil organic matter. So it's been lost from the soil, returned to the atmosphere. So we've messed up this equilibrium between the soil and the atmosphere. We're turning the atmosphere to be more in equilibrium with the soil. And so we're trying to get that carbon now back out of the atmosphere and repay that soil carbon debt. Okay, so there's been some very nice studies on this. This is Cynthia Kallenbach. They did a really elegant, simple experiment in the lab and showed that carbon use efficiency on the x-axis here is strongly correlated with soil organic carbon formation on the y-axis here. We know that the chemical properties of the carbon, the food that you feed to microbes, matter. Carbon that's more reduced, so that it has higher electron density, so that has a more negative oxidation state, results in higher carbon use efficiency. And that's basically because there's more electrons per unit carbon, and that microbes can be more energetically efficient with using that carbon than other carbons. Now, there's costs associated with that. It requires more energy to break those bonds. Microbes have to make enzymes, etc. So it's not as straightforward, but generally speaking, things like lipids and things with a lot of carbon-carbon bonds, they're more electron dense, and their use is more efficient if the microbes have the machinery to get at that carbon. And then metabolic modeling across hundreds of microbial genomes has shown that the potential carbon use efficiency, if you just consider flow of carbon through the whole metabolic network, is related to the phylogenetic origin of the organism. So that says that there's some sort of evolutionary history that's constraining carbon use efficiency. The point I'm making here is that there clearly are some evolutionarily conserved traits that can be related to carbon use efficiency. So we're going to go looking at some of those, what they are. All right. So I'm going to talk about two papers that we've published related to this. They feed off each other. I don't have time to go into the details of each of these, but you can go and look at these. But I'll give you the main take-home message. The first one here is work. This is by Kate Zalnina. And here we were able to show for the first time that, and I'll show this cartoon here, but we can start off here at number 1. But just think about a soil in which a seed has just germinated. So that soil is made up of, it's a seed bank of microbes with all sorts of different traits. Those microbes are there waiting to be activated. A plant germinates, a seed germinates, the roots start to grow, the shoots grow above ground, the roots grow below ground. As those roots grow and the plant develops over time, those roots are releasing chemicals. They're releasing sugars, amino acids, other organic acids, hormones, lots of different things. And as those roots encounter microbes, microbes are able to consume some of what the plants are releasing. Now, the plants release different chemicals as they grow over time, according to their own allocation needs. And it turns out that microbes basically follow what the plants are doing. They follow the phenology of the plant because the chemistry of the root exudates are changing over time. The types of microbes that grow with that plant are also changing over time, directly following the substrates. So the preferences of the microbes for different substrates that the plants release drives the succession of microbes to follow the development of the plant. Okay, so that's what this paper showed. We were able to read that from the genomes and predict which microbes would grow at different times of the plant development according to the types of metabolites that the plant was releasing and which we measured. So it turns out that at the beginning, the plant releases things that are sugars and amino acids, and eventually releases things that are organic acids and fatty acids. And actually, these end up, for the most part, having different carbon oxidation states, so different electron densities. And so we would expect maybe that this succession of chemicals drives a succession of microbes with different efficiencies. And we observed that slow-growing bacteria were very competitive in this environment, even though there's a lot of resources, which was unexpected. We always thought that the microbes that grew around plant roots were the weeds of the soil microbiome. They just grew and they flourished. But we had slow-growing organisms able to compete in this environment, which was surprising. So we tried to work out why that was happening. And so then Gianna took all of this information from that experiment and the traits inferred from genomes, and she used her dynamic energy budget model to try and diagnose what was happening here in this situation. And this was Nature Microbiology, I guess, the year before last. Okay. So we have the same schematic here, and I'll give you the punchline here. Basically, right at the start, when plants start growing, they're releasing sugars, and those sugars are they're right at the sweet spot, like glucose and sucrose, where they're easy to access for microbes. They got a reasonable electron density, and they enter into central metabolism, like sucrose and glucose. So these are microbes. They stimulate microbes that grow extremely fast, whereas the microbes that grow later on are consuming carbon that has higher electron density, but is a little bit harder to access, so it requires a bit more energy to access. Those microbes grow slower, and what we identified was that the slower growing bacteria using those types of substrates are more efficient. They have a preference for those more reduced electron-rich substrates, which gives them a higher assimilation yield. So the action of the plant, the chemicals that the plant releases, selects specifically for organisms that are more efficient, and the substrate itself leads to higher efficiency. So those microbes are more efficient, and the substrates that the plant is giving to them also enhance their efficiency. So that allowed those microbes to be more competitive in the environment. They're building their biomass more effectively, which allows them to outcompete other organisms for other resources like nutrients in the environment. So I'll zoom in a little bit on this here. You can see the green bars here are the organisms, the slower growing, more efficient organisms. They have a preference for amino acids and organic acids in this environment here. The main point of this, I realize there's a lot of information on this slide here, is that what explains the competitiveness of these organisms and their carbon use efficiency, the first thing is the assimilation yield, which is directly a function of the carbon oxidation state of the substrate. So the more electron-rich substrate explains a lot of the variance. But then the traits of the organism itself, its synthesis efficiency, its affinity for those substrates, its uptake rate for those substrates as well. These are all important contributors to the efficiency of that organism. So it's not just the substrate, it's the other traits of that organism that determine their ability to compete in this environment. So that suggests that organisms that are selected for by these root exudates are more efficient. And so we have the potential to be able to manipulate for example, plants to produce more of these substrates, to select for microbes that are more efficient and enhance the production of soil organic matter. So this is a to be continued story. We're working on this, and we're using calorispirometry to do this. And that's basically measuring how efficiently microbes use substrates by quantifying their heat flux as they're consuming substrates and producing CO2. And I'll leave this story here.
So question, you're
Yes.
focusing on manipulating plants. What about fertilizers, biochar, other additives?
Yep. Yeah, great question. So I would say the answer is you're using all of the above. The other dimension to this as well is that the balance of resources has to be right. If you have insufficient nitrogen or phosphorus, then the carbon use efficiency of a microbe will go down. So that's a really important part of the story also. So the stoichiometry, the ratio of elements, is extremely important, as well as the metabolic efficiency of the organism for the carbon. That's a really important component of this. I'm not showing that here, but great question. Things like biochar are very effective at absorbing and holding on to resources in soil, also manipulating the pH. And both of those can alter the rate of supply of resources to microbes. And as I mentioned at the beginning, microbes that take up resources really quickly tend to be less efficient. So if you can slow down the supply of resources to organisms in soil, you can improve the efficiency. And biochar is really good at doing that. So that's another potential method or lever to manipulate in these systems.
So like a resource battery.
Exactly. Yeah, great way of putting it. Yeah. Okay. So we can, assuming you believe what I've told you, we can predict important microbial traits directly from genomes and integrate those into models. Does this make a difference to biogeochemical fluxes if we integrate this information into complex ecosystem models? So remember, with all of those other factors interacting. And this is a project led by Virginia Rich. We collaborated on this, part of an NSF-funded eMERGE project. Jen Lee was the postdoc responsible for this work, and this is published in Nature Communications last year. And the context for this is atmospheric warming, melting permafrost. And if you're not familiar with permafrost are ice lenses that are formed at high elevations or high latitudes. With atmospheric warming, these ice lenses melt and the landscape subsides. So you get these hollows formed that become water-filled or water-saturated. Under those water-filled or water-saturated conditions, the diffusion of oxygen is limited. And as other microbes consume that oxygen with the carbon that's available, and then the diffusion rate is slower than the consumption rate. So these systems become anoxic or anaerobic. They have a lack of oxygen. And that promotes the activity of anaerobic microbes like methanogens and fermenters. Fermenters produce the raw materials that methanogens need. Methanogens take those, like hydrogen and carbon dioxide or acetate, and they produce methane. And methane is a significantly more potent greenhouse gas than carbon dioxide. So there's a very large potential for the release of massive amounts of methane from permafrost in Arctic regions that could exacerbate and feed back into enhanced global warming. So this is a major concern, and our Earth system models don't do a good job predicting this, and partly because the representation of microbes in those models is either, if it exists, it's very rudimentary. So this is the context. Microbial metabolism is a key modulator here. So we were asking the question, do microbial traits have ecosystem level consequences? Does this matter when you zoom out and you look at an entire landscape? So this is a study look above the Arctic Circle in Stordalen in Sweden. This is a permafrost thaw gradient where we're going from the dry, intact permafrost to a bog, which is where the ice lens has started to melt, and then to a fen where the ice lens has completely melted. So you're going in increasing saturation, increasing potential for methane production. So this field site has been ongoing for a number of years, where we've been with many collaborators studying the genomics of these systems, which microbes are present, turning those genomes into traits, and now integrating those traits into an ecosystem model. So this is the Genomes-to-Ecosystem framework again. We have metagenome assembled genomes. We then have the microtraits, prediction of the traits, assimilation into DEB microtrait to predict further traits. And then we're using that information then to parameterize an ecosystem scale model called EcoSIS. And I'll tell you a little bit about that. So EcoSIS represents all of these processes that you see on the right-hand side here. So it represents the physical processes of soil, water, heat, gas, and solute fluxes through the soil-atmosphere interface within the soil. It represents the dynamics of plant growth, plant resource allocation, photosynthesis. It represents organic matter decomposition by microbes, and how all of these processes are influenced by external forcings like precipitation, temperature, humidity, radiation, etc. Now, what makes EcoSys relatively unique in the sphere of ecosystem models is the depth of microbial resolution. There are 11 functional guilds. Now, from a microbial ecology perspective, that's fairly basic treatment of microbiomes. From an ecosystem model perspective, this is by far the most sophisticated, fully integrated land atmosphere model in terms of microbial biogeochemistry. So what we've tried to do is we've tried to improve the representation of these guilds of microbes using information specifically from these sites. And then we ask the question, does that make a difference? So EcoSys...
Actually, Tom, do you have a question? I think he has his hand up.
Oh, yes, please, go ahead. Tom, did you have another question?
No, I guess I was just still left up, sorry.
Thanks.
Okay, sorry.
No worries.
Thanks.
No problem. So this is using a similar Michaelis-Menten type formulation, representing the maximum specific reaction rate as a substrate, as a function of substrate concentration. And so what we're doing is we're using the microtrait and DEB microtraits to predict these parameters for each of those guilds within EcoSys. So when we do this, the gray bars here show the genome inferred traits. The white bars show what we would get from literature. Now, remember, any models that are microbe explicit have to get their parameter values from somewhere. All of those come from observations for the most. All of those come from observations. That's the white bars. Those observations are made using organisms that grow in the lab. Those organisms may come from a tropical forest. They may come from a desert. They may come from an agricultural field. They're very likely not coming from the site at which you're using that information to represent the activity of microbes in. So you're using, it would be like using parameterizing a vegetation model for the Arctic using trait parameters that are derived from plants that grow in the tropics. So there's no reason to believe, or there's a lot of uncertainty in whether those traits are representative or not of those organisms that live in these systems. So we've been trying to infer those traits directly from the genomes, from the sites where we're doing these model simulations. And you see in some cases here, like the aerobic heterotrophs, at least for the maximum specific respiration rate, the literature values and our genome inferred values agree pretty well. They don't agree well for the half saturation constant. For others, like the methanotrophs here, they don't agree well for either of those. So that means that if we were to use the literature values relative to the site-specific genome-inferred values, we're likely going to get a different answer. And so that's a warning to say that we really need better ways of if we're going to use these types of models, better ways of obtaining these parameters. And I'll be honest and I'll say that our ability to test the hypotheses that these traits are accurate is very difficult because our ability to grow these organisms is limited. Where we do have data where we have genomes and measurements, data sets where genomes and the lab measurements intersect, we do a pretty good job, but we don't know whether that holds to these organisms that have yet to be described in culture. I'll say that, so the ecosystem-specific trait values can differ very markedly from literature. All right. So when we put these, use these traits, genome-inferred traits into these models, we can then test the sensitivity of specific outputs of these models to microbial trait parameterization. And what you're looking at here is the blue line, the solid blue line, is the simulation best fit. The orange dots are the observations. These are flux chambers measuring methane flux in these systems. And then the light blue is the range around that simulation best fit. So this is, as we alter all of those microbial trait parameters, look at the variance that introduces. It's massive in terms of methane emissions. So microbial traits in these models contribute to a huge amount of uncertainty in the prediction of something as important as methane. And then we can zoom in on these the different functional groups, and on the y-axis here, this is a mean elementary effect. So what is the relative effect of manipulating each of these parameters on methane flux? And heterotrophic or hydrogenotrophic methanogens, sorry for the shorthand here, HM, these are organisms that produce methane using hydrogen and CO2. The model is extremely sensitive to their parameterization. It's extremely sensitive to the fermenters that produce the raw materials that the hydrogenotrophic methanogens need. It's also sensitive to the aerobic heterotrophs that compete for those resources. It's negatively sensitive to the methanotrophs as well, which means these are the organisms that consume methane as well. So all of that makes sense. We know that these should be the sensitive nodes in the model. What I'm not showing here is that the choice of microbial parameters also has a big effect on things like net ecosystem exchange, which is the ability of the ecosystem to absorb carbon dioxide or carbon dioxide equivalents. So plant growth and performance are also impacted by microbial parameters, not just methane flux. So the main point here is that microbes, they're important catalysts. The consequences of how we represent those in models has feedback beyond the direct products that those microbes make themselves. So it cascades through the ecosystem into things like plant growth and performance, as well as methane fluxes. So microbial traits do matter. And if you conversations with ecosystem folks who work on ecosystem and earth system modeling, I think, reasonably, the question has always been there: does this matter? Does variance at this scale matter at all? And at least in these complex models, it suggests that yes, these traits, these features of microorganisms do matter at these scales. Okay, I'm going to wrap up with this story with this. If you remember, we had those gills, and then we have a range of parameters. We have a distribution of parameters. The ecosystem model has one representative organism for each of those gills. So for every type of microbe in there, we have one value we can use with the way the ecosystem model is written at the moment. So what would we use? Would we use information from all of the genomes? What statistics would we use from the distribution? And what does that do for our predictions of methane? And basically the short version of this is, if we use all genomes present, we obviously got a wide error, much more error in prediction, and our correlation is pretty low, is 0.3 for both the two locations, the bog and the fen. If we use the 80% dominant, so assuming that just the abundant genomes matter, we still have a lower agreement with the observations and a relatively high error. We'll use community aggregated. In other words, we weight the traits based on their abundances in the genomes. And this is commonly used in vegetation modeling. We increase our agreement with the observations by about 90%. So using the community aggregated seems to be a really good approach if we have to choose just one value. Now, microbes, like plants in these ecosystems, are dynamic. As the conditions change, different types of microbes grow. That will happen seasonally as well as so both in space and in time in the soil. So in reality, the trait distributions are not a static value, they're dynamic. And we really need to be able to represent the emergence of these kinetic traits over time to improve our accuracy in predictions. The challenge is that this ecosystem model is already complex. We're talking about introducing more complexity into that by adding in more versions of microbes so that they can basically fight it out, and that the trait distributions can emerge over time based on which microbes are fittest. So that would be aligned with this community aggregated method of trait parameterization. So I'll just say very quickly, we're doing that. We have another model. I'm going to skip through this in the interest of time. We're building this new model called EcoSIM, which is a recoded version of EcoSys, which is the model we used in that permafrost example there. It's been recoded so that it's much more modular, and it can run, and it's more parallelizable and run an HPC compute. And importantly, it allows us to add new functional groups and guilds and have multiple representatives for each guild. So that way we can have different versions of the hydrogenotrophic methanogens, methanotrophs, etc., with different kinetic parameters, so that the community can assemble over time, and then the kinetic trait distributions emerge over time. We believe that will give us an improved representation of fluxes, or closer to the observations that we make. The last thing I'll say on this, when you start to do things at this scale, you have to be able to parameterize the environment that the microbes live in as well. And so as part of this project, the Watershed Function Science Focus Area, this EcoSIM model allows us to bring the landscape, the bedrock, the soils, the vegetation, all of the traits at the whole watershed, allows us to bring all of that together into one simulation framework. And we're building approaches to observe the landscape and parameterize these models using real-world observations. So we're using aircraft and satellite-based LiDAR to give us the elevation, slope, aspect, curvature, vegetation, canopy height, and structure. We're using hyperspectral data to represent the plants themselves, the canopy, that we can identify the types of plants. We could also quantify the amount of carbon, water, nitrogen in the canopy, using machine learning derivations of that along with field observations. We can predict other properties of the canopy, other traits that are important for plant function. We follow literally the aircraft in the air with geophysics. We make geophysical measurements so we can see below ground what is the soil texture. We take soil cores with that to relate that texture to chemical composition, organic matter content, and microbes. And we do genome resolved metagenomics to build in the relationship between microbial traits, soil physical chemical properties, plant landscape position, water, energy availability, where I started at the beginning. And all of this information is being built into our workflow to parameterize this EcoSIM model. So this is a work in progress, and this is my last slide. And I'm right at time, so hopefully we still have folks can hang on for a few more questions as well. Thanks.
Well, fantastic. Thank you. Jendra, you have a question?
Yeah, I have a question, two questions. One is like how we can verify our results with like we are using experimental or in-field data collection. And second question is the second question is like are we also considering the positive rhizopriming effect, negative rhizopriming effect in our modeling, so we can get the idea of stable carbon getting stable formation of stable carbon from labile carbon.
Great. Yeah, great questions. Thanks. So the first one was about how do we verify results. using either in-field or let's say experimental manipulations or lab experiments. So I'll say these modeling approaches like the EcoSIM, for example, or EcoSys, we can use those to simulate lab incubations using the exact same formulations. We just basically we change the environment to represent the vessel that the incubation is happening in. So we try and mimic the physical environment that the incubation is happening in. So that's one way we can test exactly whether this model is producing the expected results, and we can simulate exactly the conditions that the experiment is happening under. So that's one thing. We also do, I said we can only cultivate a small minority of organisms, but that's still many organisms. So we still do cultivate microbes. We read their genomes, we try and cultivate them. Sometimes we're successful, and then we test the predictions that our genomes, the assumptions of our genomes, and we test the predictions of our models. So we do lab experiments to test that. The thing is that we'll never be able to get close to the diversity of microbial life on the planet. So we have to systematically go after that diversity in our cultivation approaches. And there's other people, globally doing that. They're systematically going after the tree of life to try and isolate representatives. And that's the way to do that, and then build up a body of knowledge so that you're confident that your predictions extend beyond the narrow window of organisms that you've actually been able to, that you've actually observed. And then the priming effect. So for those that know, the priming effect is that there's a couple of different aspects to this, and that's the stable carbon in soil. That stable carbon in soil can be destabilized, and that means it can be made more accessible to microbes for metabolism when other substrates are added that are more favorable. That can happen in a couple of different ways. One is the substrates that are favorable, that there's a pH effect, and then the materials that are not accessible to microbes are desorbed off those materials and are made available to microbes. That can be represented in the model by representing pH changes and how pH influences organic matter mineral interactions. The other side of priming is where there's an energetic constraint. So let's say microbes could access this carbon, but it's going to require an investment in enzymes to be able to access this other carbon. And they don't have enough resources coming in to be able to produce those enzymes. So if you were to add in sugars and things like that into soil, microbes now have more energy and can invest that in making enzymes, and they can then access this otherwise inaccessible carbon sources through the production of enzymes. Those are two examples. I will say for both of those, the EcoSIM does a better job at representing the enzyme production component of that than it does the pH component of that, but that's a part of the code that's being developed right now. So that the surface, the organic matter mineral interactions component of that, it is present in the code. It just needs more complexity built into it. But yeah, it's possible.
Got it. Thank you very much.
Great question. Thank you. Yeah.
And then one more. So EcoSIM sounds like a relative of what Descent and those more soil chemistry focused models, but obviously with a microbial component. So what's the relationship? Which processes do you incorporate? They do not, and vice versa.
The fundamental difference between those is that EcoSys or EcoSIM are really built off first principles processes. Descent, the century line of models, are empirically based. So there's empirical formulations relating the reaction rate of a specific pool of carbon under certain conditions, so temperature, moisture, oxygen regulation, etc. So they're very empirically based models, which means they have to be parameterized for given systems. In many cases, they're not. You're using the same empirical formulation derived from experiments in the 1970s or 1980s. I'll say those when they're very well tuned, those models can do a good job because you're basically representing the statistical relationships between external factors and behaviors within a system, but you're not representing the actual processes that are happening. You're representing a statistical association. EcoSys or EcoSIM are based on theoretical formulations, are as close as possible to first principles, so that the behaviors like that you see in data, those statistical relationships, they should emerge, but you don't prescribe them. And that allows you to capture behaviors outside of conditions for which your empirical formulations have been derived, which we think is, if you're trying to predict forward in time into no analog conditions, it's going to be hard to rely on something with an empirical formulation that hasn't considered those future conditions.
Is there a trade-off of you requiring additional data measurements that in order to parameterize these?
Totally. Yeah,
and
exactly. So yeah, that is the trade-off. It is the EcoSIM series of modules. Overall, these are not all manipulable parameters, but has about 1,300 parameters or so. So your degrees of freedom is enormous, and your parameter sensitivity could be enormous also as well. Now, every one of those, though, is designed to be something that's measurable at some level or another. So I would say for microbial traits, for example, we don't want to have to go to every single site. We don't want to have to do metagenomes and assemble metagenomes. That's just not practical. So what we would like to be able to do is we build a representation of the types of organisms that might possibly exist. We have those in the model, and then we let the first principles approaches take over and select for the organisms that should be most active under those conditions so that those traits emerge, as opposed to having to prescribe them in a static way each time. So that's the goal, is that it's a dynamic it is a dynamic model. Let's say an example where you don't have to rely on that assembly over time would be in agriculture. Maybe for microbes, you might let those microbes assemble. But if you know your crop and you've got your traits measured over your crop, you know exactly which cultivar it is, you already have all of those data. So you build those data in. Those data then, based on the allocation scheme for plants, they will very strongly regulate the resources that are made available to microbes in the soil and the plant microbe interactions. But the microbes will self-assemble under that scenario, and their trait parameters will emerge over time. So I would say the cost benefit of these types of models, definitely there's a much higher burden of parameterization for these types of models. And the question is always, well, how suitable are the default parameters for this system, that I'm working in? I think over time, our strategy here, though, is to run massive ensembles of these simulations with many, many trait combinations, and to actually derive surrogate models from those using ML approaches. So that's our strategy, longer term strategy.
So then there's other maybe hybrid, like I'm thinking mimics, where they have just two kinds of bacteria, and they don't even name them. They just have two behaviors, two strategies. So how does that trade off? Really, maybe the question is the data availability. They found some data sets with soil columns to train themselves. Is that kind of data good enough, or do you need a new generation of data collection?
Yeah. Well, I think it's a different. Maybe there's a philosophical difference, but there's also a practical difference in the use of these models. So things like mimics are specifically targeted at organic matter, mineral interactions, and so representing that. EcoSIM or EcoSys or DEB are a general formulation that can be used for any application. You can do micro-mineral interactions that influence organic matter. You can also represent denitrification. You can represent nitrogen fixation. You can represent nitrification. We can build out whatever number of microbial functions matter at an ecosystem scale. And because all of these processes are intertwined and interconnected, you need a model that represents the primary pathways of energy yield and transformation and organic matter yield and transformation by microbes. So that's been our strategy, has been to build a more comprehensive model that has multiple functions, and that comes with a parameter burden for sure. So I would say something like mimics. You could reduce potentially what we observed in the rhizosphere, these fast-growing and slow-growing organisms and their influx on efficiency. You could reduce that to a simpler model that might look more like mimics. that would be, you could come up with a simple model that could be useful for that application. I think that, but we'd know why we were able to reduce it. We'd understand that process as opposed to, building up a simple model that does a good job at representing your process. If that simple model does a good job of representing your process, are you satisfied that you understand the reason why that simple model worked? I don't know. I don't want to be controversial about this because I really appreciate that there's no one model fits all, and models are really tied to your use case. The fitness of the model is really tied to your use case. So some of this comes back to the use cases that we work on, really trying to go to much larger scales, but we need to represent interactions of many different types of processes for our use cases. And so Mimics has its uses, Dascent has its uses, and EcoSIM is a multifunctional model. But we have the ability to condense EcoSIM and build reduced order models from EcoSIM as needed for specific use cases with lower parameter burden if needed. But that's not our focus at the moment.
That makes sense. Yeah, and then these are various design trade-offs along the spectrum.
Exactly.
So
yeah.
Now, if you have a bit more time, just looking at you're focusing on the kind of like Earth system modeling, or at least large-scale analysis of these dynamics. That means you have large-scale data sets.
Yep.
So what kind of data is available to parameterize this family of models, even if it's like Mimics family, but with like 10 bacterial families based on your analyses that is more theory-driven? But still, what data is available to constrain such dynamics?
Yeah, great. And I think we've tried to kind of view where the data generation trajectory is going. So we looked at particularly remote sensing. Remote sensing, Alpha Earth, for example, is just, it's potentially transformational once we understand what those embeddings mean. It really is. I mean, this is something. So we've been trying to do this with our own aircraft, commercial satellites, and ground-based sensing in our watersheds where we're working in the Colorado River. Very similar to Alpha Earth, but our pixel sizes are 50 centimeters. Alpha Earth are 10 meters. We have data layers below ground also, as well as all of the the Alpha Earth above ground. And actually, the Alpha Earth embeddings have a lot of correspondence to our clustering as well. So I think the key thing is observing the surface of the Earth system. You can derive data products from that to parameterize these models. So we can derive plant traits. We can visualize based on spectral signatures what types of plants are where. We can identify their greenness index, their proxy for chlorophyll content, their nitrogen content, etc. So that's and then by reference to a database for that type of plant, we can then pull in traits for that plant in a gridded way for any location. So we will be able to do this. Then you need to be able to connect to that. You need to be able to connect, well, what are the traits below ground that we can't see? And so what we're doing is, and other people are doing, is we're exploring relationships between what you can see on the surface and what you can't see below ground. And so at the start, at least, that requires, this theory. We can predict soil property distributions based on patterns of soil formation, position in landscape, what the soil properties might be. We have models that can already predict that. So you got some models predicting other data products from what you can see on the surface. And then we will do sampling campaigns to verify those hypotheses. But we're always learning new things. So the sampling campaigns will give us new information, refine our models, etc. There are other things like the Earth Microbiome project that has sequenced metagenomes for microbes across the world. At very sparse and not evenly distributed, of course, but we're going to have more and more and more of this. We're going to have, again, already most of our information of microbes comes from genomes. We're going to have more and more and more of this as sequencing costs continue to plummet. The quality is going to get better. Our ability to interpret those genomes is going to get better. So we're going to kind of get out ahead of all of that to make a framework that takes advantage of all of this information that will be available at large scales. So for now, to do this in a system requires lots of expensive coordinated measurements if you want to take advantage of these models. We're lucky enough to do that. We have good funding from the Department of Energy and National Science Foundation and others to help do this. And we coordinate with other research groups to work together in the same locations to try and bring this together. But eventually, these types of data sets are going to be available globally for, all of our data is open. These are going to be available for people to use openly. And we and others are building workflows to ingest this information, derive the data products, initialize the model, run the model for you, interpret the model. So, you think about where this could go with agentic workflows. It's going to be an entirely new way of approaching ecosystem models. And then the type of, version of EcoSIM or the type of model you use will depend on the question that you start off your prompt with in that case. So I think it's really exciting. It's going to take many years, still quite a few years for us to get there for all of that data availability, but clearly that's where it's going. And it's just going to make these models way more accessible to our community and to practitioners as well.
Nice. So then, if I may, there's now, in addition to the modalities you described, there's things like eddy covariance measurement that can be dragged over drones. So that's a new modality. Ground penetrating radar for much cheaper way of getting soil bulk density. There's even ecoacoustics, which seems a bit insane, but somewhat actually tells you something about the lives underground. So how can they be useful, as additional?
Totally. Yeah. I think eddy covariance theory, that's a benchmark, once you work out the footprint of that tower, and then you've got this is my colleagues run the Ameriflux network at Berkeley Lab. So, this is something that we're thinking of. In our field sites, we have eddy covariance, and, we're trying to see, can we recreate those observations? There's also as well as drones, there's other autonomous robotic platforms for sensing that can be deployed to measure things that are ephemeral. All of these things are part of a kind of a changing ecosystem of how we do this type of work. And I'm trying to remember your last question. Oh, the bioacoustics and things like that. That's information. These are signals, to be deconvoluted. What do they mean? I think that's absolutely fascinating. I give one example. Our work in the watershed, probably the most interesting and important things happen under the snowpack in the winter, but we don't know enough about that because it's hard to get there, hard to access. But that's when microbes are cranking, turning over the previous season's organic matter, releasing the nutrients, making, building their biomass that then gets released and releases nitrogen just when the snow melts and plants green up. It's this synchronization of behaviors, and we really don't know what's happening. But yet, when you clear off the snowpack, there's a thriving ecosystem right underneath that snowpack, and bioacoustics and things like that would tell you, there'll be an index of activity there. Maybe you could identify different features with different types of earthworms, things like that. Fascinating, I think, approaches to be able to bring all these sources of information together is really intriguing.
Oh, fantastic. Thank you very much. This is really valuable.
Great. Well, thanks a lot. And thank you, everybody, for joining and for your questions as well.
Thank you very much.
Yeah, have a good rest of your day.
Okay, cheers. Bye-bye.